Welcome to UT Brainstorms. I'm Mike Mock from the Department of Neuroscience at the University of Texas at Austin. Our department sponsors UT Brainstorms as a way for us to share our passion and expertise about neuroscience and a way for us to learn from you and your questions, what interests you and what concerns you. This is Brainstorms 38, the second of our 5th season. So thank you so much for your support and continuing to show interest. As always, we want to thank Laura and Elena for everything they do to make Brainstorms happen. So because Brainstorms is a conversation, the format that we use is designed to facilitate that. So after our discussion tonight, or after our talk, we'll have a question and answer period where you'll type your questions into YouTube chat. Feel free to begin typing in those questions beforehand. It lets our panelists get a chance to start looking at the questions early. So it's my pleasure to introduce tonight's speaker, Eric Senning. Eric is a part of one of the great strengths of UT Neuroscience and in fact of UT Molecular Biosciences, which is we have a particularly strong group of scientists who study ion channels, which are little gates at the surface of neurons that mediate their signaling and so much of what they do. Eric's one of our young guns in that group, and he's tremendously talented and has agreed to share his expertise tonight. He's going to talk to us about, as you'll see, a form of ion channels that helps mediate our perception of temperature and heat. And so just so that we can get things going, please join me in the virtual welcome of Dr. Eric Senning.
Hi, everybody. So I'm going to start sharing here. Screen in just a moment. There we go. All right. Well, I would like to specifically thank Dr. Mike Mock, Laura Ian, and Elena Silva for organizing the UT Brainstorm seminars, and also for inviting me to speak on the topic of the spicy brain, a conversation about how we perceive heat. And also, before I forget, I want to thank today's panelists, Dr. Lauren Dobbs, Dr. Andres Yarroz-Aguirre, and Dr. Rick Aldrich, who have all agreed to participate in a panel discussion at the end. And before I forget, I also want to thank my daughter for this nifty little drawing that she made of what she perceives of as heat, which is several different types of sensations. You know, it can be the hot stove, where the capital H is shown here, or maybe when you're lying in the hot sun, you've got an umbrella over you, and you could even be a little chili pepper. And just as a reminder that, you know, heat isn't always so great, you've got the little zingers all around the heat word here to show that, yeah, it can sometimes be a little bit dangerous. Let me just start by saying that this topic is near and dear to me because of my research on the molecular receptors that are responsible for how we perceive heat. And today's talk will highlight some of the ways that this kind of culminates in our perception of it in the brain, and even down to the molecular details of how that stimulus starts out. So when I was asked when I'd like to give this talk, I immediately thought of the fall. And well, for most people, this could mean, you know, seasonal changes and the leaves on the trees turning from green to yellow or red, or perhaps even the start of the school year. You can see some children lining up perhaps for lunch, although maybe for this particular year, it's a little different for families. It certainly has been for mine. Growing up in northern New Mexico, the fall also signifies an important agricultural event, and that is the harvest of green chilies in southern New Mexico. And here you can see that at all the grocery stores, you have these big drums lined up and roasting chilies by the pound for folks to pick up. And as a teenager, we would do this and get about 25 to 35 pounds of roasted chilies in this huge garbage bag that we would then lug home and put in Ziploc bags to store for later use in the year. But I wasn't always a green chili addict, and only became one later because I was born in this little landlocked country here in Switzerland, in Western Europe. And that place, of course, was well known for other things like mountains, and in terms of food, just really rich things like cheese and chocolate. And growing up there with my brother, of course, we didn't eat only rich food like that, but we also had our fair share of healthy snacks like bell peppers. And the reason I bring up bell peppers in my brother, here he is at the ripe old age of about 5 or 6, is that they may have had some influence on where things turned out in my life. Is that, and before I get a call from him that I showed this corny picture of him when he was 5 or 6, I can assure you that he got the better end of the bargain when it came to my grandma's haircut. So here's me alongside him there. And haircut aside, he was a pretty cute kid, and I think he knew this because it really came in handy for his practical joking. He's about the biggest practical joker I know. So when my mother's cooking took a turn towards Indian cuisine, his innocent offer at one time to have a bell pepper snack, when he came out of the kitchen, I pounced on it, not knowing that he had actually carefully prepared some really spicy peppers by removing all the seeds like this and laid out some of these chili peppers in that plate. And I, of course, unwittingly ate some and thought I'd learned my lesson there. And I did for a little while. Of course, it wasn't long before we went and visited a sushi restaurant. And with my brother around, I learned of all the myriad ways that you can hide wasabi in sushi rolls. So these were my early experiences with the spicy peppers, and may have also affected the trajectory of spicy food in my life. But let me quickly outline today's talk so that you have some idea of where we'll start and maybe end up. And that is, we'll start in the brain. Well, of course, this is a UT brainstorm, so it makes sense to look at how sensory input is integrated in that seat of cognition. And then we'll sort of reach out and look at the receptors and nociceptors that provide input to the brain. And then we'll look at one particular type of ion channel that is really crucial to how we sense temperature and pain. And it's also called the capsaicin receptor for a specific reason that it responds to capsaicin. And finally, we'll talk a little bit about how it seems that some of us really acquire this taste for spicy food when it really ought to be the opposite, that we should try to stay away from it, since it is kind of unpleasant the first few times you experience it. And full disclosure here, I am now a spicy food addict. You can ask my kids, they always see me put spicy sauces on things when they just don't understand why, but maybe they will in a few years. So let's dive right in here and talk about the brain, since this is a brainstorms talk. And what I've sort of always marveled at in looking at the brain is how much of the brain is actually devoted to sensory input. Here you have a depiction of the cortex, which is sort of the larger part that you see on the surface of the brain here. And it's shown here with the outline of a head to give you some orientation. And there's parts of it that are, large parts that are dedicated to processing of vision, processing of touch. In the frontal lobes, you have processing of smell, and then the temporal lobes, hearing, and even taste. There's processing of the insular cortex, which is a little bit buried here. But a lot of the cortex is devoted to how this stuff is acquired from peripheral senses and then integrated in the seat of where we understand things. And another aspect of sensory information that I find so alluring is that there are some experiments that can be done that are really non-invasive and makes it easily accessible to look at how we perceive the stimuli that come from the outside world. And this is the discipline of psychophysics, relating stimuli we have to our perceptions. And a lot of fun can come from this with even simple experiments you can do with young children and adults alike. And I've listed here a link that we'll put into the chat at some point later on, where you can find different types of experiments that are a lot of fun to engage in. And one of these is called the 2-point touch discrimination test. And what you do here is you get some index cards and you tape down some toothpicks at different distances along them. And you see at what points in the body one can still discriminate how far apart those toothpicks are. And you'll find out, through methods of randomly trying these cards in different places, that the fingers, the upper lips, and the cheek are really quite good at telling apart when there are two toothpicks present at very close distances. And this is not at all the case of other parts of the body, like the back or the shoulder, where these toothpicks have to be much further apart in order for the body to sense that there are, in fact, 2 points being touched there. What's astonishing about this simple discrimination experiment is it correlates very closely with the actual inputs of the sensory information going to the somatosensory cortex. And so from neurological studies, we know that the inputs of these, the sensory information into the cortex, where it begins to get processed down into information about how much, intensity, and exactly where, that this surface is not at all in a one-to-one relationship with the surface area of the body. And just like that 2-point touch discrimination assay showed, there are regions that really get overemphasized for somatosensory information, again, like the lips and the fingers and the hands, and other places where there's really poor representation up here, like the calf, or again, the back. And even, modern philosophers like René Descartes was sort of aware of this unusual property in somatosensory input, in that he noticed that, for instance, that people who were blind would pick up sticks and use them to gather information about objects in the world around them. And so with the loss of vision as a sensation, one could adapt another aspect or modality of sensation to provide representation in terms of the world around us and give us information about it. And even today, people who are visually impaired and use the white cane will also use it to discriminate objects around them and build up a representation of environment. But even information about like the hardness of an object, for instance, if the cane is tapping against asphalt or turf or dirt, can be felt through just minute vibrations felt in the cane going up through the very sophisticated sensory apparatus in our hands to inform us about what it is that's in front of us or around us. So let's have a look at how the central nervous system actually gets this information to it. And here's sort of a rough breakdown of 2 components that are important here. We have the central nervous system that comprises the brain and the spinal cord here. And then we have the peripheral nervous system. And this, gets the information starting here in the head from afferents that innervate different tissues in our mouth and our cheeks and even parts of our eyes to convey it through cranial nerves into the brain. But then a large component of information from our extremities and from our organs even is through peripheral nerves that innervate our skin and extremities. And these branch out at different points along the spine in a very stratified and predictable sequence. And that sequence can be sort of mapped out to different zones along our skin according to where these nerves branch out along the spine. And you can see here, for instance, that in this figure of a person here on the left, the green region are nerves that are, innervating as they come out of the spine along the cervical vertebrae, and then going down towards the waist, you have the thoracic vertebrae and afferents that are extending from these into different zones of tissue here. And this continues along towards the base of the spine and towards our feet in a very predictable pattern. So let's have a look at the anatomy of these nerves that are extending down from the brain out to our extremities. Pardon me. So here it illustrates, this figure that the 3 sort of the typical 3 neurons that gather information about our extremities and convey it along through the spine and up to our cortex to begin processing in the central nervous system. And at the very periphery, we have what are called the primary sensory neurons. These are typically along the spine dorsal root ganglia. This is the dorsal root here along this branch, and they extend out into the periphery, gathering information and being sensitive to deflections here or temperature here. And these synapse then to a secondary neuron that's located within the spine, and it ascends from there up towards the brain, where there's another synapse in the thalamus, which acts sort of like a relay station for most of the sensory input going into our body, I think with the exception of olfactory sensation. And finally, a tertiary neuron carries that information, the final step for processing in the cortex. Well, what I said about this pathway isn't quite true for all somatosensory information. In fact, there's another pathway shown here on the left, which is called the dorsal column system, where again, you have a tract going into the spine where a dorsal root ganglion is sort of the primary neuron. And then rather than traveling across the spine, as I showed you before, it goes up the dorsal column on the same side of the spine. Turns out that information from this sort of tract is almost exclusively fine touch and proprioception. Proprioception is how we maintain balance in our body. Typically, these are nerves that innervate our organs and our muscles. And it crosses over here in the brainstem before continuing on to the thalamus into that tertiary neuron. On the other hand, you had, as was shown in the previous slide, the neuron synapse and cross over to the opposite side of the spine before climbing up into the brain. So this can actually make for some very unusual circumstances if there's an injury in the spine around the nape of the neck. In this example here, they've shown a lesion or a hemisection of the spinal cord on one side. And the consequences to that would be that for the same side, there'd be loss of this fine touch sensation and proprioception again, because you lose here this point here of connection. But at the same time, on the opposite side, you lose sense of pain and temperature sensation because that tract is traveling along the opposite side up here where it gets cut off. And consequently, this individual here, for instance, might feel pain on one side, but not really know where it could be localized to on this red side of their body. So this would be sort of very unusual experiences of pain and touch as a result of an injury in the spinal cord. The relay of this information along 3 neurons also poses some interesting questions, allows one to pose some interesting questions about whether or not these are just a direct signal relay to the brain and the brain does all the work trying to translate what goes on, or whether or not there are circuits in place here that already sort of refine the information and filter it to have a specific output. In this first example, we look at a hot stimulus, right? And this might indicate something that's painful so as to avoid it. And the type of neuron would pick this up is one that's tuned to heat. I'm gonna take a quick drink here. That activity from this neuron is then passed on through a synapse into the secondary neuron, a spinothalamic tract neuron, which is then going to pass it on to a thalamocortical neuron, the tertiary neuron, and of course, finally received in the cortex and appreciated as a painful sensation. For cold, it's slightly more complicated. Turns out there's 2 types of neurons that are active when there's cold. One that's sort of mildly active and it interfaces with a specific type of spinothalamic tract neuron, which we've colored red here. And then when it's integrated into the last neuron, it actually has an inhibiting role. So it's actually suppressing the activity of this last neuron. On the other hand, for cold, there's a very sensitive neuron that is twice as active when it's cold. And it conveys that high degree of activity to this intermediate neuron, the spinothalamic one. And then when this information is integrated, you have basically 2 minus 1, which is still 1 and suggests that yes, there still would be pain here because it's cold. However, when things are cool and not necessarily painful, now the activity of this blue or dark purple neuron is actually less. And you can see that the way this is integrated through one side inhibiting and one that's activating is that no, there's no pain in the end because this is only cool and not cold. Now, it turns out that around the turn of the 1900s, Thunberg did some very interesting experiments with mixing these modalities of hot and cold and discovered that if you introduce a sensation of warm and cool at the same time, you can actually induce painful sensations. And this was shown sort of nicely with this apparatus here, where you have slats that are alternating hot and cold across the surface of the hand. And this was repeated again more recently. And what appears to be happening is that you activate some of these hot neurons with the warm grill, which is set to about 40 degrees. And when these then synapse onto these red type of neurons that also get information from these light blue neurons, they inhibit the activity of the cool neurons. And so you get no net function or no net activity of this red neuron on the thalamocortical neuron. But on the same token, you have these cold neurons, these dark blue ones, still conveying that activity through the green one. And when that's integrated at the end, overall, there is a painful stimulus that is experienced. And that's shown here in this study with subjects indicating that with warm grow, where all the slats are warm, it feels hot, but none of them reported any pain. All of it was cool. They all reported it was cold. But if you did this alternating hot and cold slats, many of the individuals in the study said, hey, this actually doesn't feel very good. And I've heard it described as sort of a prickling sensation. Another atypical pattern that's looked at is with mechanical stimuli. So if you introduce mechanical stimuli, they also seem to suppress the activity at this second neuron here, so that you can overall experience a painful sensation. And the nearest I can think of how this might be is if you were to grab onto a already cold, let's see, metal pole, the pressure of squeezing against it with your hand would be a mechanical stimulus, and it would still be cool. And so perhaps you'd feel a tingling sensation there. Whereas if you were just holding onto a cool pole that eventually got cold as you held onto it, so you're cooling it, while at the same time not inducing any mechanical stimulus, you would just notice that it's cooler than usual. So this is sort of some of the cool stuff that happens as these neurons convey information on the ascending pathways. And I think at this point, it's worth looking at the anatomy a little bit of those primary neurons that are gathering information from the extremities and the environment. So in this sort of classical depiction that I'm showing here, you have maybe a typical neuron you find in the central nervous system that has dendrites splayed out around the cell body. And these carry information like depolarizations from these processes, extensions, and then might, after these are integrated, the axon hillock cause an action potential to form, which is then carried on down to presynaptic sites where a synapse will convey that through chemical neurotransmitters to the next neuron here, which then causes this one to fire an action potential and carry that information further again. Now, it so happens that the way that these afferents and primary neurons at the extremities are formed don't quite have their soma out at the extremities, and that's because the field from which they have to gather their information is actually much larger than what is typical for a central neuron. On top of that, there are, of course, injuries that can happen here at the extremities where one wouldn't necessarily want to lose the whole neuron. One would perhaps just allows this thing to grow back its axon and rebranch out here to form new interactions with the tissue at the distal site. And these still collect information and can depolarize out here to fire an action potential along this leg. And that's carried on then to central sites in the spinal cord, where these then cause the secondary neurons to raise their activity due to the action potential being carried over. So what do these neurons look like if we characterize the endings? And we can see that depending on the modality of somatosensory input, they have all sorts of different shapes and interesting bodies and anatomy. So there are these Merkel disc type touch receptors that end in these little bulbous bodies that detect touch, or they're the free nerve endings that are very susceptible to temperature changes, and these are temperature receptors. Then there are also these unusual ones that wrap around hair follicles and can sense if the hair follicle is bending, and these are susceptible to stroking of the skin where hairs are bent to the side, or different sort of corpuscles. And some of these are pressure receptor or touch, or in this case, one of these is actually receptive to very high frequency vibrations. And some of these are located deep in the tissue. An interesting class of these are the free nerve ending receptors that innervate not just the dermis, but all the way out into the epidermis, where there's no longer any blood flow. And these are also the pain receptors that are very crucial in sensing when there's about to be an insult to the tissue, and therefore allow an individual to avoid it. I think that some of the greatest work to show how unique these endings are comes from microscopy imaging that's been done with different fluorescence markers for these different types of neurons. And here in panel A, what you can see is that one of these neurons that has these Merkel discs at the end has these unique little bodies, and then these go into a myelinated neuron that forms the afferent that then carries information and signal back to the cell body. Just below it here, you actually have a hair follicle with one of these circumferential type nerve endings going around it, but there are also these very unusually spiky shapes here. And this is a different type of neuron called a lanceolate. The neuromantic tracing that's done here can show this even better, where the circumferential type neuron ending is shown in blue, and the lanceolate ending is shown here in white. And these sense fundamentally different things. The circumferential receptors are sensitive to stroking of the skin, and these lanceolate ones are actually susceptible to indentation. They sense when the hair is being pressed down. So they sense different modalities of touch. In this figure 2, you can also see some of the Merkel cell bodies here connected to an afferent that's then conveying information about pressure and touch. And here are many follicles with these little lassos around them, ready to sense stroking of the hair follicles. And here's some more lanceolate endings to sense the indentation pressed against the skin. Some interesting work to show the density of some of these nerve endings is done in work with even human skin tissue. And this is most likely from a biopsy that was taken from an individual, where you can see below the basement membrane, you have blood vessels and tissue where blood supply provides all the nutrients here. But then beyond that, in the epidermis, there is no longer any blood flow, but yet these nerve endings persist all the way out there, remaining functional just through diffusion of nutrients along these very, very fine fibers that are sensitive to things like temperature changes and pain. And of course, if you look in the tissue of someone who's suffering from a type of peripheral neuropathy, you can see that the density is much reduced of these nerve endings. And that might lead to pathological conditions where there's increased sensitization to pain here because the nerve endings are no longer responding correctly to the stimulus that they should perceive as innocuous. All right, where else do you see some of these very fine nerve endings? And one tissue that's still epithelial is the cornea, which of course is something you really want to make sure you protect as being your eye. And in the zoomed in region here, C of this box, A, you can see how highly innervated this region is with these small, thin afferents. And in some cases, it looks almost mesmerizing how these trail around in these radial patterns. It's been very useful to do this fluorescence microscopy where you can label specifically molecular markers for these different afferents. And in this case, TRPM8 is going to be the ion channel that's responsible for cold sensation. So this is a cold sensing neuron here that's wrapped around. And CGRP is a marker for nociceptors. So you can see how many of these very thin afferents are actually going to be the receptors that sense pain. So I think I've mentioned nociceptors here, and I should talk a little bit about what they are. We know from studying these tissues in samples from mice and rats that there are various sizes of diameter fibers that run these tracts out to the periphery as primary afferents. And it turns out the thickest fibers are myelinated. They have sheaths of myelin that are quite thick. And these are responsible, again, for proprioception, which is our sense of balance, as well as light touch, so high frequency vibration. This is carried through these A alpha and A beta fibers. Then these thinner myelinated ones are now allowing for nociception, so they sense mechanical pain thermal and chemical pain as well. And finally, for the unmyelinated fibers, these come from small diameter neurons, and they sense innocuous temperature, right? Because they're very thin, they can be sensitive to temperature in the surrounding tissues, not insulated, as well as itch. And of course, there are nociceptors, again, as before, with mechanical sensitivity, thermal and chemical sensitivity. For nociceptors to work and be helpful to us, they have to have an activity threshold that's just below tissue damage, allowing an individual or animal to just get out of the way if things are going to be damaging. And they innervate almost every tissue in the body. There's, of course, a notable exception, and that's the brain. Well, that might actually make sense, because if there's damage to the brain, there might not be any hope at all for the animal to survive. But in tissues where it seems very important for the animal to learn a lesson, they're, of course, highly innervated. And this could be regions like tooth pulp, where a person or an animal might want to know, yeah, you should save your teeth. They're important to you. They help you survive and thrive. As well as the cornea, where you want to protect your eyes, because vision is really helpful for you in the long run. And as I said before, most painful temperature sensations are carried by these unmyelinated nerves, these C fibers that can innervate the epithelial tissue that's really at the most distal sites in our body, in our skin tissue. So the work that's been done to look at these different types of neurons came a long way when one realized one could extract these peripheral neurons and prepare them in culture. So these here would be the dorsal root ganglia that are collected from the side of the spine. And we can use molecular markers to image which ones of these would be interesting to look at with electrophysiological techniques. And in this sample here, the small diameter neuron is one that's going to be a nociceptor type and sensitive to temperature, and not the large diameter one next to it. So one could identify the small diameter ones as unmyelinated and therefore interesting to pursue in studies of a temperature-dependent activity. So here on the right, you can see a typical electrophysiological recording of an unmyelinated small diameter neuron, where if you apply heat, there are these large currents in the membrane due to ion channels opening in response to this modality of activation. Similarly, it was found that there is a agonist called capsaicin, that this molecule, when applied in pretty low concentration dosages, would similarly activate many, if not all, the neurons that were heat activated. And so this became a very useful tool in looking at which neurons would be responsive to heat, because it works as a functional analog to that. One can also look at activity from a action potential perspective and looking at the voltages. So at the lowest concentrations of capsaicin, when puffed gently against a neuron, one wouldn't see any activity from it here. But if one started to increase the concentration to 10 nanomolar, these puffs would elicit action potentials, and even higher concentrations, there would be more and more action potentials. And these kind of experiments here are the bread and butter of my work. And I thought maybe there's a way to share this kind of information with you in another modality than just looking at traces like these. After all, we're talking about sensory systems here, and maybe there's a way to also put this forward in a sensory modality that's not quite just looking at the data. And it turns out there is a scientific tradition of doing this in astronomy, where signals that are time varying can be detected with large array apparatus here that look in space for radio waves. And these are then converted into audio tracks and played over a speaker system. And so if you allow me, I'm going to just a second here, go back to pointer mode and play you a track of a pulsar that is being watched in time. So yeah, I thought that was pretty interesting. So why not try the same thing with what we're doing, looking at these neurons that are activated by this molecule called capsaicin. So we have here an example where we patched onto a cell, and here we're going to now flow over the cell of capsaicin and watch the electrical activity, but also record this electrical activity and provide it to an amplifier and see what we hear when we play this. So get ready, and you're going to hear perfusion lines change, and then you won't miss the activity of this neuron.
Womp bom balloon bom 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 to the boot, oh root, 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 oh womp bom balloon bom 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 bom, I gotta get.
Yeah, of course. Maybe a little bit of my brother rubbed off on me too with his antics. And of course, neurons don't sing like Little Richard if you put capsaicin on them. But nevertheless, it is an actual recording here shown below. And one of the interesting characteristics of this neuron is that even though we activate it with capsaicin, eventually this neuron will sort of suppress its firing and calm down a little bit and not carry on like Little Richard can for the rest of that song. But we'll come back to them in a minute. Let's talk a little bit about this molecule called capsaicin that does such interesting things to heat-sensitive neurons. So here's the molecule shown as a molecular structure. And it has this phenyl group here, which is attached then to a long carbon chain that makes it a really greasy substance that likes to hang out in oils and in lipids, most commonly are lipid plasma membranes. And it reacts and interacts, with the TRPV1 receptor, which of course we'll talk about too, is responsible for the pungency of sensing, spiciness in hot chili peppers. And we have to wonder, like, why would chili peppers necessarily make this thing that's so offensive? And it turns out there's some benefit of having it that protects these plants from microbial infection. But by far and away, the most important thing it does is it discourages seed predators from eating these peppers. So mice and other mammals will steer clear of it because it affects them by binding this heat receptor and making them steer clear in a way. But this hypothesis that it only discourages seed predators isn't complete, because it turns out that birds that also have this receptor TRPV1 have a slightly modified version that do not taste the spiciness. And so they consume the pepper and the seeds, and it turns out their beak will preserve the seed. And so when they carry it away and their droppings land really far away from the plant, it can actually be somewhat beneficial for the pepper because it's a seed dispersal system for them, spreading the peppers much further away. So one of the most unusual things about this idea that there's a directed deterrence is that it doesn't really work for people. In fact, what I have here on the left shows what lengths people have gone through to raise peppers that are even spicier than the naturally occurring ones that we have around. And this is the Scoville heat scale, which is based on a very subjective method of testing spiciness. So at the bottom here, we have a simple bell pepper, and the scale goes up in units of dilution. So going up the scale to the jalapeno, for example, it's 2,500 times more diluted before you get the sense that there's no spiciness in this in this dilution. And you can go up the scale here to something like a habanero, and that's where I cut out. I really am not a big fan of things going beyond a habanero, but enthusiasts have gone all the way up to something like the Carolina Reaper, which is cultivated by someone on the East Coast and is, I think, considered to be one of the spiciest hybrid peppers that you can find these days. And to celebrate, these sort of peppers, there's these big festivals where people gather around and participate in these competitions where I can only imagine that the pain they must experience getting to some of these peppers is just unbelievable. But what you'll notice in front of them is these emergency cups of milk. And the milk is really helpful here because it's loaded with fats and lipids that when you put this in your mouth and you rinse out your oral cavity with it, it'll carry away a lot of that capsaicin because it's such a greasy thing that prefers to reside in the milk than stay in your tissues in your mouth. So that's a helpful tip there on that regard. capsaicin on the research side has been really helpful to us and scientists, because it allows us to do some cloning techniques and expression cloning and work with nociceptors to identify what were the molecular components responsible for heat activity. And it was then recognized that this TRPV1 protein, which is an ion channel, is responsible for being the site of activity for capsaicin, and that there seemed to be almost exclusively heat-activated nociceptors and cold-activated nociceptors that had a different type of ion channel responding to cold. And once both of these were cloned and identified through gene sequencing, one could then look back with antibody labeling at the ganglia, which are these clusters of the neurons that reside next to the spine. And through labeling, see that there's almost this mutually exclusive labeling for these two types of receptors. Here you can see the red receptors, of course, are heat receptors labeled with red, and the cool receptors contain tryptophan are green. So once we have this ion channel cloned, we can then work with it in heterologous cell systems. That means expressing it in non-neuronal type cells and just functionally analyzing them, how they behave. So here they still have the same functional behavior as one sees in nociceptors responsive to heat, as well as to capsaicin. It definitely has TRPV1 in it, this neuron here. And one could also look at how they respond to different temperature ranges. And we can see that this is VR1 here, is exactly the same as TRPV1. It was just renamed more recently. And at about 40 degrees, which is just above body temperature, we see this steep rise in activity, which is right where this receptor would want to become functional if it was responsible for nociceptive behavior in our primary afferents. One could also then make a knockout mouse of TRPV1, and you can see that they were very successful in this regard, because here you can see this little guy is happily munching on a very spicy habanero, where I'm sure all his wild type litter mates are, cowering on the other side of the box. I can't believe he has his whole snout braid in there. That seems like it would be unimaginably painful if he still had a functional TRPV1 channel, but he doesn't. Other pursuits with TRPV1 were to identify its sort of molecular structure. And we can see here on the right that this was done, and it shows that the channel is made up of a tetrameric arrangement of identical subunits. And if we turn it on its side, we can even look down the pore in the center, and it's surrounded by very characteristic voltage-gated potassium channel type structure, even though this channel turns out not to be very voltage-gated. The pore is non-selective, allowing both calcium and sodium to go into the cell. And we know from its functional studies that it's activated at temperatures greater than about 43 degrees, also by capsaicin, of course, and modalities like acidity, so low pH, as well as sulfur organic compounds and even toxins. There are some very potent spider venoms that activate this channel. And of course, in a mammal that would be experiencing this kind of toxin, it's sort of like a very searing hot sensation that the toxin induces in nociceptors that contain TRPV1. Some other interesting properties about TRPV1 activation is that over time, there seems to be sort of a desensitization effect. So here one can see a current that's shown across a nociceptor that contains TRPV1 because it's responding to capsaicin. But over time, you can see that the signal is getting smaller and smaller and weaker as repeated activations can come in. And this is a lot like what we saw with the neuron that labeled little Richard, in that you can't sustain activity with these channels open all the time. And this turns out to be due to a calcium-dependent effect in the neuron. Another interesting aspect highlighting the importance of TRPV1 in health and medicine is that if you, in this area called thermal hyperalgesia, and what this really means is an increased sensitization to inflammatory pain. And you can see here, if I try to explain this figure from the right side first, that if we look at wild type mice, so these are ones that have a fully functional TRPV1, one can place these mice on a hot pad and note how long it takes them before they lift their little feet up, because it's about 50 degrees or something, so not too pleasant. But if we add or inject them with an inflammatory compound called carrageenan, it takes them much less time before they lift up their feet, because now their foot pads are inflamed, and so they very rapidly lift up their feet. It turns out in the heterozygote, which still has one functional TRPV1, the behavior is much the same as before. But if we look in the full knockout mouse of TRPV1, now there's really no difference before the injection of carrageenan and after. And so it seems that TRPV1 has this central role in inflammatory pain. And this can be highlighted here in this figure, where in this primary afferent, which is clearly a nociceptor containing TRPV1, and now seeing increased activity due to tissue injury around it, where it's releasing this compound called CGRP and substance P into its environment. And it's also being regulated and modulated by neurotrophins and other metabolic factors that are in this inflammatory soup around it, which is both acidity, ATP, or serotonin, and bradykinin. Central to all of these different influencing factors is TRPV1, which is modulated in some way by all these different compounds. So one can see that TRPV1 is very likely to be an important tool to look at in terms of treating pain. And this was known already for some time. In this paper that's more recent, one could see that injecting mice with a large dose of capsaicin would make them tolerate this sort of hot plate assay for much, much longer than in animals that were not injected with capsaicin. You can see that their delay before lifting their little feet is relatively short compared to one that's been desensitized with a large dose of capsaicin. And this actually sustained itself for long periods of time. So many days later, these animals are still not lifting their feet pad up off the plate. And one can also look at this in the context of inflammation with this compound called CFA, where we have, first of all, some of the mice that were injected many days before, continuing not to notice how the heat pain from this hot plate. But at the same time, one can look at mice that start out noticing it because they haven't been treated yet. But in the same day where they're given this injection for inflammatory measures, some of them are also injected with capsaicin. And so the ones that are injected with capsaicin on the same day go right up to the level of the animals that no longer lift up their feet. Whereas the ones that were not injected, they respond much like they would with just a mouse that's on this hot pad here, but also having some inflammatory pain at the same time. And what this has led to, of course, is that you can buy capsaicin topical ointments over the counter in pharmacies. And here, for instance, you can see a patch that one can purchase for treating, localized pain to the back, or even creams that are a formulation of capsaicin up to 0.1%. And I think with a prescription, it's possible to get very, very high formulations. And searching the internet for some of the information on this, I also found this helpful side effects of capsaicin cream guide here, which says, of course, it causes pain and inflammation, and I would not recommend putting it anywhere near your mouth. But I found it odd here on the right side. It suggested that there's these concerns about it, being on your hands, and yet here you have this individual modeling how to apply it with their hands to their arm. And as anyone can tell you, in my family, we still get green chili and then get them ready for storing them in the freezer. You always wear gloves when you're handling lots and lots of green chilies with capsaicin on them, getting them ready for putting them in the freezer. Maybe this is just a secret of people who live in the Southwest, but of course, PPE is a good measure to use against capsaicin. I heartily recommend it. So rather than just using capsaicin to desensitize TRPV1 in these neurons that are innervating our skin and acting as nociceptors, what about just blocking these channels? I mean, that's a thought. So there are some potent antagonists of TRPV1 that have made it through the research pipeline to early phases of drug trials. And the problem is that when these are introduced to a model animal like a mouse, it turns out that their abdominal temperature changes and it actually becomes somewhat higher. And you can see here there's a raised temperature in the mouse. So they develop a hyperthermia. And this was very specific to the channel TRPV1 because you can see it only affects animals that still have a functional TRPV1 receptor and not the mutants. And if one goes back and looks at the potent agonists of TRPV1, so like capsaicin, there's another agonist called RTX. It's even more powerful. If you inject an animal with that, you got the converse effect here. Now the animal's experiencing hypothermia as its body temperature plummets. And looking at a list of different drug trials that were done over time and the effects on body core temperature, you can see that both capsaicin and RTX have this effect of inducing hypothermia. And most of the antagonists that they've attempted to use induce hypothermia in model systems. And even in one case, this was tried out in human and revealed that it was still inducing hyperthermia in some of the drug trials that were done with people. And you might ask, well, that doesn't make a lot of sense. Why would you allow such a drug with such a big side effect to continue? And it has to do with the fact that there's a really strong drive to develop new pain treatment strategies to compete with the current paradigm of pain treatment, which goes to using opioids. So I think that there's still a good reason to look at TRPV1 as a target for treating pain. It's just going to be a lot harder than one initially had thought. But in terms of thermoregulation, I think these TRP channels also highlight something really curious in that there seems to be a balance between those receptors in our peripheral nervous system that are detecting temperature that's hot or cold. And you can see here along the bottom that if we introduce these TRPV1 antagonists, we develop hyperthermia. And then if we use agonists for these other cold sensing neurons, we actually also induce a hyperthermia. While on the opposite side, if we introduce an agonist for TRPV1, then we have a hyperthermic state. And coming back to what it could be about these the use of capsaicin that might be rewarding or interesting to people, I'm not sure it's the sweating that one looks forward to, but perhaps something that lowers your metabolic rate can have some sort of associated reward. But I'm not so sure. What we do know is that some of these ascending neurons that are secondary neurons from nociceptors also interact at the parabrachial nucleus in the brainstem with a neuron that carries a signal to the amygdala. And this is a central site in the brain for processing our emotions and also how we develop aversive behaviors to things that are bad. So sort of a fear. And that makes sense because my experience with peppers a long time ago taught me not to accept anything from my brother anymore, but also to stay away and be wary of any kind of red peppers that were lying around, even though they might also be bell peppers. I couldn't be too sure. At the same time, there have been some studies to suggest that capsaicin can also raise the endogenous endorphins in your body. And in this study, a group injected different types of agonists for nociceptors, so capsaicin or generic formalin, which is going to introduce inflammation, and found that with capsaicin, you had this increase in the beta endorphins in your body, and these bind to the opioid receptors and induce sort of a very rewarding experience. Although I'm not so sure that anyone it's enough peppers to have this much capsaicin in their body, it would be interesting to maybe talk about this as we go into our final stage today and start the panel discussion. So with that, I hope that everyone found this an interesting talk and learned something about pain receptors. I really want to thank University of Texas for my time here so far and the many wonderful people that I've worked with in my lab, as well as the labs that also study ion channels that I know really well, the Gold Genome Lab and the Ulrich Lab. Of course, the organizers for today's Brainstorms, Dr. Mike Mock, Laura Nian, and Elena Silva, and collaborators and support while I've been here. So with that, I think we're going to move on to the second stage. I'm going to stop sharing and introduce our panelists today. So I have Dr. Andres Yara Osguera, who's with us from the Department of Molecular Biosciences, Dr. Lauren Dobbs, who's with us from the University of Texas Neuroscience, and Dr. Rick Aldrich, also from the University of Texas Neuroscience. And well, I think we're ready for your questions.
Eric, great job. Thank you. Welcome to the panelists. Please now start typing in your questions. I see that they're starting to appear, and I'll be funneling to our panelists. So Antoine asks, is this process similar in the tongue's perception of Szechuan peppercorns?
I'm going to take a short break here because my throat. Would somebody else like to address that?
Hi. Yeah, so I am not 100% sure, but I remember having seen a paper where they actually.
Hey, Andres, I'm sorry to interrupt, but we can barely hear you.
Oh, okay. Maybe I will try to speak louder. So I remember having seen a paper where they actually tested that, and it seems that that molecule is also an agonist of TRPV1. So probably the mechanism would be similar, although the level of activation and desensitization that it causes could still be different. So it could be the same in some ways and different in others.
I think as long as you isolate capsaicin from whatever's in those peppers, then it can be a sure bet that it's activating TRPV1.
Okay, so this question is from Joe Virgil, who's a longtime Brainstorms enthusiast and always has great questions. How might a brain injury or brain tumor affect a person's ability to sense heat and pain?
Well, I could sort of start a preliminary way of answering that. I'm not a specialist about the central nervous system, but anytime we're looking at something that interrupts those tracts that convey the information from the periphery up through the thalamus and then into the cortex, one could probably or conceivably see an effect in sensing pain in some way. And then, of course, in the cortex itself, that parietal lobe is really crucial for processing that information. So it's sort of going to be probably a regional effect that has to be along where those tracts run. That's my best guess. Maybe somebody could answer who knows more specifically about the CNS about that.
I'll say I don't know much about it, but I was reading some papers in preparation for this panel and, talking about capsaicin and how it can actually act as basically an analgesic, right? As you were saying. And some people mapping out kind of the central anti-nociceptive circuit, starting with the striatum, the nucleus accumbens, and then descending down, through the periaqueductal gray, and then the rostroventral medulla, and down into the dorsal horn. So I would imagine perhaps maybe not its pain if it's centrally located, the tumor or insult is in the brain, but it could affect maybe the anti-nociceptive or the analgesic properties of the capsaicin.
So here perhaps is a similar question. The question is there any way that people who are congenitally insensitive to pain, is it possible that they're missing the TRPV1 receptor?
Yeah, I could take that one. There are people like this, but the mutation is in a different ion channel, and it's a sodium channel that's involved in the transmission of the pain signal up the spinal cord. So it's not down at the periphery where Eric's talking about the TRP channels being, but it's for a specific type of sodium channel that's involved in the ability of the signal to get on up to the brain. There are other there are drugs that also act on these spinal cord pathways. There's one that's used for it's called Zycomatide, which is a calcium channel blocker that actually, instead of coming from chili peppers, comes from cone snails. And it's used to treat pain in opiate-resistant terminal cancer patients. And it's a calcium channel blocker. It works really well, but it has to be pumped directly into the spinal fluid, because if it's administered by a shot or nose spray or a pill, then it blocks too many of the tight calcium channels, and it affects regulation of blood pressure in a really dangerous way. So it limits its ability to be used for pain treatment. But it's another example of something that works on the pathway and not on the beginning signal in the periphery.
Sorry, I got sidetracked by 3 different questions at once here. So there's a quick question here about why is it that usually children are more sensitive to spicy food than adults?
Well, I can take part of that. If so, one reason might be that so actually, if you eat a lot of chili pepper, you can end up killing the terminals that detect it. And so you build resistance essentially because you don't no longer have the nerve terminals that can detect the pain. So if you are an adult and have been eating chili pepper all your life, that could be one mechanism. I am not sure if there are any other developmental regulation of these channels. So that is definitely one.
But what I've always found so curious is why would it sustain itself, right? I mean, you could take it up to that level where you've desensitized yourself to capsaicin, and then, you need more and more and more of it. And what is it that's driving people who are, enthusiasts, maybe not quite like myself, but even more so, to keep wanting to get more spice, right? I don't really have the answer to that one. I mean, maybe there is some kind of reward mechanism there that, that's through a different way than just the peripheral nociceptors, but maybe more central ones. I don't know. TRPV1 is expressed in other tissues than just in the periphery. So perhaps it's acting at sites like that.
There's a theory that I think is kind of anecdotal, but the idea is that people that live in hot climates tend to like spicy food, and the hypothesis is that it makes them sweat so they cool off better.
So it's kind of a negative reinforcement argument for why you would want to eat spicy foods, because it's alleviating something else that's unpleasant, right? If you're hot and you eat something spicy, it's going to alleviate your, overheating perhaps. But yeah, we could talk more about addiction too.
Yeah, that's a great.
There are TRPV1 channels in the reward pathway of the brain, and activation of them can increase dopamine neuron firing and dopamine release in the striatum. And you can also get, basically, you can shut down, or not shut down, but you can inhibit the long-term depression of a striatal circuit, a circuit in the reward pathway that inhibits behaviors. And it can bias towards more of this, what we call the go pathway, which ends up, it's more related to drug-seeking behaviors and kind of compulsive-like behaviors. So, I mean, it's not, we're not sure that's how spicy peppers are working, but there's definitely, mechanisms there that can affect the functioning of the reward pathway and these pathways that get you to do things again and again. So maybe it's a learned behavior, right? You start at a young age and then you develop synaptic plasticities in these go pathways, and you just want to eat peppers for the rest of your life.
I mean, I really like the notion that it could be that you're alleviating something else that you find unpleasant. Why do we eat really spicy food in the Southwest in the summer when it might be a really hot day? And it is perhaps just to cool us off for a bit of a comfort. I mean, what do you think that the concentration of capsaicin in your blood would have to be in order to raise the level of capsaicin in those central circuits? It would have to be like an acute shot of it into your bloodstream.
Imagine it'd have to be pretty high. I don't know. I tried to actually find that, like how much capsaicin, based off a systemic, injection or, consumption of it, how much crosses the blood-brain barrier and what's the concentration? I don't know. I don't know if anybody really knows that.
I once went to a meeting of capsaicin researchers, which I'm not. I went because it was at a really good curry house. But the two things I learned from that was this curry place made you sign a waiver if you got the hottest type, saying you wouldn't hold them accountable for it. But the more interesting thing is that I was sitting with a woman who was in a group that did testing of chemical variations of the capsaicin structure. And they said that when they would come up and synthesize a new variant, they're sort of back of the envelope initial calibration for how hot it seemed was just to drop a little drop on their tongue. And they stopped doing that when one of the people in their lab group actually lost all sensation to anything that was even slightly spicy, like garlic or ginger or something. And I said, how long did this last? And she said, well, it's been 1.5 years and it still hadn't come back.
Oh no.
Well, here's I'm going to jump ahead. Here's a question that's maybe the opposite side of the same coin. So are there genetic mutations that make certain peoples more and less sensitive to spicy food?
I think there is a cluster, as I recall, of individuals that I think through family relations, and I can't remember if they're from someplace in South America, that have a mutation in TRPV1. And Andres, maybe you know the specifics of this or remember. I think it's somewhere along the region where the TRPV1 receptor interacts with capsaicin, that there's a mutation there that makes them less sensitive to spicy compounds. I don't recall if they're less sensitive in terms of temperature sensation, though. I believe it was specific to capsaicin.
So I think everybody knows that the better part of our sense of taste is actually smell. And so Harvey asks, do olfactory neurons play a role in sensing spicy or hot foods?
I think it's additive, that the olfactory effects don't necessarily come from the same compounds, but certainly, strong horseradish or mustard or wasabi have an olfactory component in the negative reaction to it. But I don't believe that's through capsaicin receptors, but it's some other noxious pathway on the olfactory receptors.
I think it's important to remember that the primary afferents that are innervating our face through the trigeminal neuron really go through so many different tissues that it's why something like pepper spray is so effective against individuals, that it can really cause so much response at once from such a broad region of our face and even our oral cavity, that it might be hard to distinguish whether there are specific sites in our nasal cavity that are just the smell receptors that are responding versus other innervations from the trigeminal neuron that are responding. And I think as far as olfactory neurons are concerned, I think they typically just express those GPCRs that are there to sense those compounds that are volatile. And perhaps wasabi is one that can be sensed through a olfactory neuron. I mean, there are other receptors that sense wasabi too.
I think it sounds interesting to imagine smelling heat. form of synesthesia. So I delayed this question because I don't know if we have anybody on the panel who knows anything about structure or function of ion channels, but Ty asked, does the TRPV1 have a specific heat sensitive region in the channel?
I'll let Andres address that one.
So the answer is we don't know, but so it definitely has. There have been good data showing that it depends on the channel because you can purify it and put it in different types of artificially made lipid membranes, and the channel still retains temperature sensitivity. So it's something happening on the channel. The thing is we don't know, and the main problem is that there are many regions that seem to be important. So people have done mutations or substituted parts of the channel with similar parts of channels that are not temperature sensitive. And that affects, for example, activation by heat without affecting activation by chili pepper, because chili pepper binds in a very defined place that people have identified. And so one of the proposals for explaining this is that perhaps different regions dispersed throughout the entire protein are somehow involved in detecting temperature, and that could be something very interesting, but also difficult to identify because you would need a method that allows you to examine every part of the channel, and that is hard to do.
And unlike a neurotransmitter binding site, heat is generally delocalized over the entire channel. It doesn't just bind to one part of it. So the idea that the entire channel is sensing the temperature is a tenable one in contrast to localized binding sites or voltage sensors in those types of channels.
There have been a fair number of sort of red herrings where people have made one mutation and seen some sort of change in the temperature sensitivity and assumed, well, this must be, at this point where the mutation was made must be the site. But it turns out, like Andres said, there could be many different places that affect that around the ion channel. And that might speak more to this sort of delocalized effect too.
So I have a question about temperature sensation. So I'm wondering how it is that we sense different temperatures and how it is that hot and cold are both temperatures. How is it that we sense hot and cold, and how do we sense different temperatures?
Yeah, that's a great question. And I had to, learn a little bit about that too with this talk, because as you saw that sort of the molecular specificity and the primary afferents is such that there seem to be like a subset of neurons that are sort of hot sensitive with TRPV1, and they activated some threshold. And then there's another, cold sensitive neuron that activates it at another sensitive threshold. And I think that it's a combination of sort of these specific afferents at the end, and then some sort of combinatorial evaluation of the data that comes in through the secondary and tertiary neurons up into the cortex, and for their processing that allows the brain to sort of figure out, okay, we've got like a lot of these kind of hot ones and maybe some of these. And so if we sort of integrate that information, we get a temperature that's about like this. I'm really not an expert about how that happens in the cortex or in those spinothalamic circuits, but my feeling is that it must be sort of a combinatorial effect on the way that ascends up and into the brain.
Yeah, it's so there is one interesting point there that when people started cloning these strip channels, they found that they could activate at different ranges of temperature. So some of them activated at very, very high temperatures, and people thought, those must be the heat sensor, the pain sensors. And though some activated at kind of mild temperatures, and other ones at cold temperatures. And it turns out that it's not so simple, because many of the heat sensors are just pain sensors. And for example, for detecting mild warmth, you actually need the cold sensor. And it's the the decrease in the activity of the cold activated channel that is ultimately required for you to detect warmth. So it's, as Eric said, it's a combinatorial mechanism that depends on the calculations that the neuronal circuits do, but also has to do with sensitivities of the different channels.
In some respects, I mean, I think an example I came across in the review of this is it's a bit like color vision, that, you know, you can get a spectrum of optical colors just through 3 kinds of cones. And as long as those are intact, you know, we have a full spectrum of colors that, you know, cover the rainbow. And so like that, we have a full spectrum of temperature sensation with maybe just, I mean, 2 or 3 or different type of specific neurons that can have a sensitivity range, and not requiring, you know, one neuron that fires off exactly at 55 and one that's exactly 50 and one that's exactly 45.
It's interesting that heat sensitive channels are activated by capsaicin and it feels hot. Cold sensitive channels are activated by menthol, which feels kind of cold. And both of those, menthol and capsaicin, are both used in topical ointments for pain.
So one other comment related to that is, so even the afferents, the heat ones detect their firing depends on the absolute temperature, but the cold ones actually detect temperature change. So there is some kind of computation that occurs even at the level of the afferent terminals that is different in heat and cold sensing.
Yeah, if you can desensitize with menthol too, perhaps we're just waiting for the next formulation of capsaicin plus menthol creams to hit the pharmacy shelves. Once they've reached the maximum amount of capsaicin that you could put in there.
Here's a question about wasabi, which I'm very curious to hear the answer of. So Ralph asks, why does wasabi affect some people more than others, and some feel head pain? And I'd like to add on to Ralph's question, why is wasabi unusual that the heat sensation doesn't last as long as, say, a pepper?
Yeah, so the wasabi is going to activate a different type of trip channel called TRPA1. And that sense of, you know, sort of spiciness is conveyed by that other type of neuron, which can also be a nociceptor. And I think it's just some of the properties of that neuron are fundamentally different, that the channel does not respond the same way as TRPA1 does to extensive activity. But also possibly because the compound that activates it, you know, tends to get degraded. Maybe Andres, you know more about the sulfur organics and whether they last very long or they just tend to, you know, get processed quickly, unlike capsaicin, which could hang around in the plasma membrane.
And also, so they are essentially covalent agonists. So they modify chemically the cysteines of the channel, and so they probably react with a lot of other things that are in the cells and get their effective concentration goes down very quickly.
There's a question about, is capsaicin found in other plants besides peppers?
I actually don't know the answer to that. Andres, do you know if something like a capsaicinoid is found outside the capsicum family?
No, I don't know. It's an interesting question. So the RTX that Eric mentioned is made by some kind of cactus, but it's a very different molecule too than capsaicin. So exactly capsaicin by other plants, I don't know.
Yeah, the resiniferous toxin is from succulents, actually. Euphorbia cactus, if anybody has heard of those, they make this resiniferous toxin, which is much more potent.
Do you guys know of any food that actually incorporates the RTX as a spice? No, I've never heard of one either.
Okay, so we're moving close to the end of our time. And so I'd like to do something that we've started doing on Brainstorms, which I think we find fun, which is we're going to go through each of our 4 panelists. And we've got about 6 minutes left. So that's a minute and a half for each of you. And the challenge to you is tell us something about this topic that you want to say that nobody's asked a question about. So, Andre, who wants to go first? You get a minute and a half to tell us something fun.
I'll start. All right. So I was interested in the role of these TRPV1 channels in addiction, and because I study addiction, and it turns out that people have investigated this, and ideally, because I said, these TRPV1 channels are expressed in several regions of our brain's reward pathway. And so it looks like, for some of the animal literature, that if you give a TRPV1 antagonist, you can actually decrease the rewarding value of some drugs of abuse, like cocaine, and even opiates. And there's some hypotheses about how this could work, right? Because maybe it's decreasing dopamine neuron firing, which we know is really important for us to get up and go seek those drugs and have that compulsive seeking behavior again and again. As Eric had mentioned, these capsaicin can also increase beta endorphins. This is a peptide in your brain that acts in the reward center as well to have these kind of feel good sensation. So there's some molecular evidence suggesting that, TRPV1 channels could even modulate the rewarding effects of drugs of abuse. And some people have looked at, they were interested in kind of that negative reinforcement hypothesis, right? So if pain might be a reason why people go on to abuse opioid drugs. And the thinking was that if you give capsaicin, since it can have these analgesic effects, could that actually serve to increase drug taking, right? So could this be alleviating some sort of pain that you're having and increase drug seeking? Unfortunately, the one study I found that talked about this didn't find any positive results. So they gave capsaicin to rats who were trained to self-administer morphine, I think it was fentanyl and heroin, and it didn't actually have any effects on attenuating their intake. So it might be a player, but I think it's still a little too soon to call it good.
Okay, Laura, since you brought up the topic, Harold asks, is spicy food addictive?
Yeah, I think Eric was interested in that too. It's why he brought me on the panel. I don't know. I mean, I feel like there's evidence for its ability to act centrally in those pathways. So it could. We know it can induce long-term depression of that no-go pathway in the striatum and bias towards a go pathway. So possibly, I mean, it could. I don't know how much capsaicin you would have to eat to get the brain levels high enough to do this, though, because these are all, acute brain slice experiments where there's bath applying capsaicin onto the brain. That's a great question.
I think that you do develop tolerance, though, and that's why some people can eat the really hot peppers. Is they work their ways up to that. I
think you can get sensitization, though, too. I used to eat a lot of spicy foods when I was younger, and now I can't tolerate it like I used to. And I was telling Eric, it gives me hiccups. So now if I eat something, me too.
Yes. I've always had that. That's been a bioassay for spiciness in my house.
Oh, that only started in my 30s. It just started giving me hiccups.
Yeah. That's something we could study, maybe.
Yeah.
All right, who's next?
Okay, I can go. So there was one thing I thought I have always found very interesting, and I'm kind of mesmerized by it, and is that it makes sense to have a pain receptor in the skin and in the eyes because we need to perceive the external world. But the capsaicin receptor is also everywhere inside of us, in the length of the axon that goes all the way from the skin towards the spinal cord, and it's even expressed in the cell bodies of the neurons that are there in the dorsal root ganglion. So what the function of those channels is, it's very unclear to me and to everyone, I guess. And one of the expressions that they are definitely doing something important is what Eric told you about the fact that when you inhibit the channels, you develop hyperthermia, and when you activate them, you develop hypothermia. And it has to do probably with some of these channels that are inside the body, innervating even the viscera and other parts. And they are performing functions there that are probably very different than the ones they do in the skin. And it's something that, yeah, we don't know much about. And I think it's important because it's something that kind of like having a pain time bomb inside of you that can get triggered at any point. So why do you have it?
Well, so the TRP family of channels is very large. It has many, many different genes and many different types of channels. And they're used for a lot of other things too. Not just temperature sensation and spicy food and things like that, but they're also involved in other sensory pathways. And in fact, they were first discovered in visual sensation in flies. And the TRP actually comes from transient receptor potential. And you can put an electrode on an insect eye, shine light in it, and you can record receptor potential, which is just a voltage change that normally comes on and stays on during the whole time of the light. And so the mutation was made, and often that's what you do with flies, is that you make mutations and see how they turn out. And so this was in a lab that studied the visual system, and what they found out is that the receptor potential that they could record from the eyes didn't stay on the entire time, that it was transient, that it turned on just at the beginning and turned back off again. And so that gave the name transient receptor potential, which has nothing to do with heat sensation or anything like that, but it was the founding member of this giant family that's involved in all sorts of sensation and probably in other internal regulatory mechanisms throughout the body.
Eric, you want to finish this off?
Sure, yeah. I think that what Andres said was pretty interesting, and I'm going to tag on a little bit with my own take on what I find so fascinating about these peripheral neurons that express these channels that are sensitive to chemical cues or heat sensation. And one of the fascinating aspects of these neurons at the periphery is that they're not postsynaptic to anything. So they don't form what are the canonical synapses that accumulate the machinery for chemical neurotransmitter to come open and cross. They have to somehow direct, possibly through some kind of mechanism that I don't know about yet, these sensory entities, these ion channels, out to the periphery, to the end of those peripheral axons, and keep them there, somehow keep them there so that they can do their job and sense what in our environment needs to happen. So that's one area that I'm really excited about, and I think it sort of exemplifies like what could be unusual or perhaps going wrong in our peripheral neurons as they sense the environment and the world around them.
Okay, folks, great. Thank you so much. So everybody, just please imagine everybody clapping. Such a great job, and so unsatisfying to end with silence, but everybody would be clapping if we were all in the same room. So thank you, Eric, Senning, fantastic job. Thanks to our panelists, Andres, Lauren, and Rick. Thanks so much for joining, fantastic time. I just want to remind everybody that there are more. You can go to utbrainstorms.com to see the brainstorms coming up in months to follow. And I want to remind everyone that if, as always, is that if you have questions or if you think of questions that you wish you'd asked, just email me. You can find my email address on the UT Brainstorm site. And if I can't answer your question, I will forward it to somebody who can, like our great experts for tonight. So thanks to the audience. Thanks everybody. And we'll see everybody at the next Brainstorm.